Welcome back to BioOne Education. We are diving into one of the most important chapter in your NCERT, respiration in plants. Now I know what you are thinking. Plants breathe. Yes, they do. And by the end of this episode, you will know exactly how a simple glucose molecule gets breakdown step by step to produce the energy currency of life, ATP. Let's get started. First, let us clear a very common confusion. Many students think photosynthesis gives plants energy. Not exactly. Photosynthesis makes food. It stores energy inside glucose molecules. But to actually use the energy for growth, repair, transport, plants need to break glucose down. That is respiration. Think of it like this. Photosynthesis is like earning money and putting in bank. Respiration is like withdrawing that money and spending it. And here is the most important part. Photosynthesis only happens during the day with sunlight. But respiration, it happens 24 hours, 7 days a week, day and night, non-stop. Look at the two questions on screen. They are basically the reverse of each other. Now, before we go deep into the biochemistry, let me give you the big picture. Cellular respiration happens in three major stages. Stage 1, glycolysis. This happens in the cytoplasm. Glucose gets split into two molecules of pyruvate. Stage 2, the Krebs cycle, also called TCA cycle. This happens inside the mitochondria matrix. Here, the carbon atoms are stripped away as CO2 and electron carriers like NADH are loaded up. Stage 3, the electron transport chain or ETC. This is on the inner mitochondrial membrane. This is where the real ATP factory is. Those loaded NADH and FADH2 molecule finally cash in their electron to make tons of ATP. Each stage feeds into the next like a relay race. Keep this flow in your head. Glycolysis, Krebs cycle, electron transport chain. Now let us start with glycolysis. Glycolysis literally means sugar splitting. Glyco means sugar, lysis means splitting. It has two phases. Phase 1 is the energy investment phase. You actually spend 2 ATP to activate glucose and convert it into fructose 1,6 bisphosphate. Then this 6 carbon sugar split into 3 carbon molecule called glycerol 3 phosphate. Phase 2 is the energy payoff. Each glycerol 3 phosphate get oxidized NAD plus picks up electron to become NADH and the substrate level phosphorylation produces 4 ATP. But remember, we spent 2 at the start, so net gain is only 2 ATP plus 2 NADH plus 2 pyruvate. This is the bottom line shown on the screen. Let me highlight some neat critical facts about glycolysis. First, location is cytoplasm. Many students mistakenly say mitochondria. No, glycolysis is the only stage that happens outside the mitochondria. Second, it was discovered by Emden, Meyerhoff and Parnas. That is why it is called EMP pathway. Third, and this is a favorite trick question. Glycolysis does not need oxygen. It is common to both aerobic and anaerobic respiration. Whether oxygen is present or not, glycolysis happens first. The key regulatory enzyme is phosphofructokinase (PFK). This is the committed step. Once PFK acts, the cell is committed to completing glycolysis. And remember, 10 enzymatic steps total, but NCERT will not ask you to memorize all 10. Focus on the input, output, and location. Now, watch this animation carefully. See how glucose enter gets phosphorylated, split into three carbon molecule, then each one gets converted step by step into pyruvate. The key thing to notice is the flow: investment at the top, payoff at the bottom. 2 ATP go in, 4 come out, net 2. 2 NADH are produced, 2 pyruvate come out at the end, ready for next stage. Let that visual stick in your mind. Now, what happens if oxygen is not available? The pyruvate cannot enter the Krebs cycle. Instead, it goes through fermentation. There are two types. On the left, alcoholic fermentation. This is what yeast does. Pyruvate first loses a CO2 become acetaldehyde, that is pyruvate decarboxylase working. Then acetaldehyde picks up electron from NADH to become ethanol, that is alcohol dehydrogenase. End products ethanol plus CO2. On the right, lactic acid fermentation. Lactic acid fermentation. Here, pyruvate directly picks up electron from NADH and become lactic acid. No CO2 is released. This happens in our muscle during intense exercise and in some bacteria. The critical need points at the bottom. Both types yield only 2 ATP from glycolysis step. No more ATP is made during fermentation itself. NCERT call this incomplete oxidation. Let me do a quick side-by-side comparison. Alcoholic fermentation, yeast and some plants gives ethanol plus CO2, uses two enzymes, pyruvate decarboxylase and alcohol dehydrogenase. Real life bread raising, wine and beer making, lactic acid fermentation, some bacteria and animal muscles give lactic acid with no CO2, uses lactate dehydrogenase. Real life curd fermentation, that burning feeling in muscle after sprinting, both give only 2 ATP. The purpose of fermentation is not to make more ATP, it is so regenerate NAD plus so that glycolysis can keep running. Without NAD plus, glycolysis would stop completely. Now, assuming oxygen is available, pyruvate does not go to fermentation. Instead, it enters the mitochondria and goes through pyruvate oxidation, also called the link reaction. This is the gateway between glycolysis and Krebs cycle. What happens? Pyruvate loses one carbon as CO2, that is decarboxylation. The remaining two carbon fragment join with coenzyme A to form acetyl CoA and one NAD plus become NADH. The enzyme responsible is multi-enzyme complex called pyruvate dehydrogenase. Per glucose we get 2 acetyl CoA, 2 CO2 and 2 NADH. This step is irreversible. Once pyruvate become acetyl CoA, there is no going back to glucose. Now acetyl CoA enters the Krebs cycle, named after Ernst Krebs, also called the TCA cycle, citric acid cycle. Acetyl CoA, which is a 2-carbon compound, joins with oxaloacetate, a 4-carbon compound, to form citrate, a 6-carbon compound. Then, through a series of reactions, carbons are removed as CO2, electrons are harvested by NAD plus and FAD, and eventually oxaloacetate is generated to accept the next acetyl CoA. Per ton you get 3 NADH, 1 FADH2 and 1 GTP, which equals to 1 ATP and 2 CO2. Since each glucose gives 2 acetyl CoA, the cycle turns twice. So, per glucose 6 NADH, 2 FADH, 2 GTP and 4 CO2. Those CO2 molecules, that is what you exhale. Watch this animation as each intermediate lights up. Acetyl CoA enters, citrate forms, then isocitrate, then alpha ketoglutarate, releasing CO2 and NADH at each step. Then, a succinyl CoA produce GTP. Succinate gives FADH2, and finally we come back to oxaloacetate, ready for the next turn. The cycle is the heart of aerobic respiration. All those NADH and FADH2 molecules are like loaded batteries waiting to be discharged in the next stage. And now we arrive at the main event, electron transport chain. This is where most of the ATPs are actually made. Look at the diagram. The ETC sits on the inner mitochondrial membrane and has four protein complexes. NADH donate its electron to complex 1, FADH donate to complex 2. Notice it enter later. So it produces fewer ATP. Ubiquitinone or UQ is mobile carrier that shuttle electron from complex 1 to 2 and 3. Cytochrome C then carries electron to complex 4, the final complex, where oxygen is the final electron acceptor. Oxygen picks up electron and hydrogen ion to form the water. This is why we need to breathe oxygen. It is the final electron acceptor in the chain. As electron flows through complex 1, 3 and 4, hydrogen ions are pumped across the membrane. This creates concentration gradient. Watch the electron waterfall animation. See how electron flow from complex to complex, dropping in energy at each step. That energy is used to pump hydrogen plus ion. By end, oxygen accept the electron and we get water. The whole system is like a waterfall. Electrons cascade down. The energy from each drop is captured to pump proton. Beautifully designed of nature. How ATP synthase works. Now those pumped hydrogen plus ion create a huge concentration difference across the inner membrane. This is called proton motive force or electrochemical gradient. The hydrogen plus ion want to flow back to the matrix, but the membrane is impermeable. The only channel available is ATP synthase, also called complex 5 or F0 F1 particle. When hydrogen plus ion flow through ATP synthase, it literally spins like a molecular turbine. That spinning energy drives ADP plus inorganic phosphate together to form ATP. This entire process, electron transport chain plus chemiosmosis, is called oxidative phosphorylation. Peter Michael proposed this chemiosmotic hypothesis and won the Nobel Prize in 1978. NEET loves asking about this. Let us now do the full accounting. Glycolysis gives us 2 ATP directly plus 2 NADH. Each NADH is worth 3 ATP in electron transport chain. So glycolysis total is 8 ATP equivalents. The link reaction gives 2 NADH, that is another 6 ATP. The Krebs cycle gives 2 GTP directly, 6 NADH and 2 FADH. Each FADH is worth 2 ATP. So Krebs cycle total is 2 plus 18, 4 equal to 24 ATP. Weight, I should correct, 2 GTP plus 6 times 3 NADH plus 2 times 2 FADH equal 2 plus 18 is equal 4 equal 24. But sum of the NADH from pyruvate oxidation. Let me use the table. Grand total 36 ATP per glucose. Some textbooks say 38. The difference depends on which shuttle system carries the cytoplasmic NADH into mitochondria. NCERT says 36 ATP, so go with that from your exam. Amphibolic pathway. Here is a concept that NCERT specifically mentioned the need to sometimes ask the amphibolic nature of respiration. What does amphibolic mean? It means the respiratory pathway is both catabolic and anabolic. On the catabolic side, it breaks down carbohydrate, fat, protein for energy. But on the anabolic side, the intermediate of glycolysis and Krebs cycles are used to build new molecules. For example, alpha ketoglutarate can be used to make amino acids. Acetyl CoA can be used for fatty acid synthesis. Oxaloacetate can be used for making other organic acids. So, the respiratory pathway is not just one way breakdown street, it is a metabolic crossroads where breakdown and building both happen. That is why it is called an amphibolic pathway. Respiratory quotient. Last major concept. Respiratory quotient or RQ. The formula is simple. CO2 evolved divided by O2 consumed. But the value changed depending on what substrate is being respired. For carbohydrate, RQ is exactly 1 because glucose uses 6 O2 and produces 6 CO2. The ratio is 1. For fat, RQ is less than 1, around zero point 0.7. Fats have lot of hydrogen atoms, so they need more oxygen for complete oxidation. For protein, RQ is about zero point 0.8, variable depending on amino acid composition. For organic acid like oxalic acid, RQ is greater than 1. These molecules are already partially oxidized, so they need less O2 relative to CO2 produced. Oxalic acid has an RQ4, and for anaerobic respiration, RQ is infinity because CO2 is released but no oxygen is consumed. Denominator is 0. NEET loves RQ quotients. Remember, carbs equal 1, fats less than 1, organic acid more than 1. Let me give you some powerful memory tricks. First, remember the order of stages with GLKE. Glycolysis, link reaction, Krebs cycle, electron transport chain. For Krebs cycle, produce per turn, remember 3112, that is 3 NADH, 1 FADH2, 1 GTP, 2 CO2. Only glycolysis happen in the cytoplasm, everything else inside the mitochondria. NCERT uses the exact phrase incomplete oxidation for fermentation. Use these exact words in your exam. RQ trick. Carbs 1, fats less than 1, organic acid more than 1, anaerobic infinitive. Oxidative phosphorylation is electron transport chain plus chemiosmosis, not just electron transport chain alone. This is a common mistake. Hidden NCERT fact: succinate dehydrogenase is the only Krebs cycle enzyme that bound to inner mitochondrial membrane. All other are in the matrix. And ATP synthase is also called complex 5 or F0 F1 particle. Let us quickly solve the NEET pattern question MCQ. Net gain of ATP during glycolysis. Answer is B, 2 ATP. Remember, 4 are generated but 2 are used in the investment phase. Assertion reason. A RQ of fat less than 1 R says fats have more hydrogen and need more too. Both are true and R correctly explains A. Match the following: glycolysis is cytoplasm, Krebs is matrix, electron transport chain is inner membrane, 1B, 2C, 3a statement based, which is correct. Option B. Fermentation produces 38 ATP, that is absolutely wrong. Fermentation gives only 2 ATP from glycolysis. 38 is the number for complete aerobic respiration, that is classic trap quotient. And that is a wrap for episode 8. Today we trace the complete journey of glucose molecule from glycolysis in the cytoplasm through the link reaction and Krebs cycle in the mitochondria matrix, all the way to the electron transport chain on the inner membrane. We saw how 36 molecules are produced from the just one glucose. We also covered fermentation, amphibolic nature of respiration, and the respiratory quotient. Thank you.